Hi there, and thank you for joining the Change I Am Possible, which is India's first future tech meets sustainability podcast. I'm your host, Eddie Ivel, and this happens to be the first episode of 2022. So, Happy New Year to you and your loved ones. And I thought that I should take this opportunity and have a conversation with my listeners because there's so much awesome and exciting things were happening with frontier technologies, and together we can make a difference. So I nudge you and I request you to let me know which guests you would like to see next, what questions you would like to be answered, and what can be done to grow India's future tech and sustainability movement. How can we enable the deep tech startup community? How can we create an ecosystem that's sustainable? What can be done to bring all the stakeholders together and create an ecosystem that benefits India? So do share your views and thoughts, because together we can make a difference and create a cohesive ecosystem for future tech and sustainability here in India. So Happy New Year to you once again, and do not forget to subscribe, support, share, and ring the bell, because we cannot do this without you.
Today I'm delighted and honored to have with me Mr. Paul Mola, who's the president, CEO, and co-founder at Roswell Biotechnologies. They have built the world's first molecular electronic chip to digitize biology. He received the Life Technologies Game Changer Award, the company's highest honor, for his accomplishments, which included establishing the National Genome Sequencing Program for the government of Saudi Arabia. So, Mr. Mola, it's a complete pleasure and honor to have you on the show. So why don't we start with a brief introduction, your background, and your entrepreneurial journey? Because in the tech world and even the biotech space, there is not much representation when it comes to colored people.
Thanks, Eddie. It's a pleasure to have to be on this podcast, and I applaud the work you're doing to just share the story and the good work going on around the world. So by nature, since a kid, I was a tinkerer. I like to open up electronic equipment and play around with circuits and just was never happy with the status quo. And so that is really what led me to where I am today. Having been in the industry for over 2 decades, I had actually observed the kind of tools that we are using for diagnostics or for measuring sort of biological interactions were pretty archaic. And by that, I mean is we were relying on technologies that relied on the microscope, which is a 1660 invention, to actually detect and sense these molecules interacting. And so part of what my role was to really drive, and my passion, by the way, was to drive adoption of these technologies to medicine where the impact can be felt, right? And so through some of these projects I did, like you mentioned, the Saudi Arabia Genome Project, which was really meant to drive adoption of precision medicine at a government level, we realized that the tools and technologies we were using were not sufficient. So this is what led me then to start Roswell, again, at the heart of it, driven by someone who's very impatient with when things are not okay or right, and wanting to drive towards solutions. And that's what led us here.
I think it all starts with curiosity. And you said you started your journey tinkering around with that. And then, you found your calling. You've been sort of like, pioneering the space when it comes to DNA sequencing. And before we get into your Roswell technology and the ME chip that you're building, I mean, I'm going to try and would want you to like simplify things over here, because the audience I have is largely students and the curious lot who want to know what's happening around the world, when it comes to biotech. So can you kind of like first explain what do you mean by precision medicine?
Today, when you go to the doctor, when you go to the pharmacy and you buy a pill, that pill was developed through a process where they actually did a clinical trial. The clinical trial and the selection of the people in the clinical trial has a lot of implications on whether that pill will work for you. The question is, was the clinical trial conducted by people like you? And if not, then how do we guarantee that you have a good reaction to the pill? And so this is the problem when we do sort of development or do sort of an assessment based on a population. And when you get treated by a doctor, a lot of the information is being derived from a population scale, and then you get your treatment based on that. They extrapolate it to you. The question is, we need to get to a point where your treatment is based or precise to you. And the big foundation for precision medicine is your genome, your individual genome being used to inform treatment of you. And so this is why having and pushing down the cost of sequencing to a point that it enables each and everyone to have their genome as part of their clinical record is so important, because it unlocks precision medicine where that information can be used to tailor your medications. It's not only used to tailor treatment and drugs for you, but it also can predict about disease. We can know that, okay, look, you have this gene that is a risk factor for disease X or disease Y. Maybe you have BRCA, which is a risk factor for breast cancer. And then you can start to manage that proactively versus wait to be sick and be caught off guard.
So you're saying if we sequence our genome, we can go directly and understand what is the problem with that individual and create a medicine basis on that individual's need, rather than having something which is extremely broad.
Yes, so that is the future, where you'll have tailored therapeutics to the individual. And also, when you look at when I talked about the clinical trials, the issue there, which is what I know that many governments are driving for this, is to have more representation in clinical trials, right? For example, we want more Blacks represented in clinical trials, or more colored people represented in clinical trials. The point is that if you have that, then you have at least an understanding of how this drug will work with a certain community, right? Within a certain community. But the future is that you want to get to a point where, yes, you can have tailored medication for real, where a drug is developed just for an individual of one, or a family, or a population, and it's cost effective to do that.
The human genome was sequenced in 2003, and when it was done, there was a lot of excitement. People claimed at least eventually we'll be able to eradicate all diseases after sequencing or understanding a human genome. But then a new team called Telomeres to Telomeres Consortium has claimed that the entire genome was not sequenced, because there was a remaining 8%, which was a lot of repeats, is what they have managed to sequence. So finally, we have finished sequencing the entire human genome. What does this knowledge of sequencing an entire human genome sequence mean for a healthcare? How do you think this will transform our healthcare?
What you're speaking to is a little bit a function of the inadequacies of the technology, where part of your genome has this, what we call repeats, and the letters repeat so often. And the way we sequence right now is we sequence small pieces of information, and then we stitch it together. The problem is, if you have so many repeats, how do you stitch it back together, right? So that becomes a bit of a complexity. And it's taken us time to actually understand that. In fact, many of the tools we use today, there's no single technology really does it well. And you usually need to combine information from different technologies to get a complete picture of your genome. Now, what happened with the whole Human Genome Project was they wanted to create a reference genome, right? Which means this is the first complete genome of a human being sequenced. Now, when they sequence you and they sequence me, then what we do is we compare our sequence to this reference genome. This is how we do it today, right? So it's almost like you have a copy and you're making a photocopy, and then you compare the two to see where the difference is. That is kind of how we sequence. We need to get to a point where we can actually sequence what we call de novo, which means I don't need a copy. I can just go directly and read my genome, right? This can only happen with newer tools and newer technologies. As long as we rely on sort of the older sort of approach, this is where sort of there's a bit of a gap. And the point though is we have a good reference genome that we are using to understand a lot of biology. But now the next step is we need to have our own genomes. You need yours, I need mine. Everybody needs their own, and it's going to be on our medical records. That's the leap that is yet to happen, right? And we need to move, and that for that to happen, the cost needs to come down, and it not only below $100 per genome, but the technology that is being used to derive that genome cannot be $1 million, right? The technology today is very expensive, and then you need to pay $1,000 to get your genome, and that's the bottleneck. So we need to go to a point where it's $100 on a device that is very low cost, that can sit in all the hospitals and all the clinics and enable us globally around the world, each and every one of us, to have our genome.
What do you think will take us over there? Is Roswell ME Chip the answer to something like that?
The answer became very clear when, in my role at LifeTech, when I was handling portfolio strategy and looking at our roadmap for technology. What the thought leaders said is, look at the communication industry, look at the computer industry, look what the chip has done for those industries, right? And do the same for biology. Put biology on a chip. That is the disruption that will unlock this future. There was a day, I don't know, I love photography, and it was a hobby of mine. It was very technical. You had to buy films, you had to go manually shoot the photos, and you had to, do all the settings manually, and you will take the photos in, and hopefully when you pick up the prints, that the photography worked okay. You will never know. And yet today, because of the microchip, I can pull out my phone, I can focus, and all the adjustments are done automatically. And immediately before I take the photo, I know what the photo is going to look like. This is the power of the chip. Suddenly, I don't have to rely on 24 prints, or my film has only 36. I can take as many photos as I want on a device that I have in my pocket. This is what we're trying to do. We're trying to put all biological information at your fingertips, at the fingertips of medical practitioners, fingertips of researchers, so that they can have comprehensive information they can use then to inform your health, to inform your well-being, to make these amazing discoveries, to drive us to a point where you can have a drug tailored for you, because it's very fast to do it. It's very cost effective. It is exactly the same revolution we expect to see in biology as we've seen now in other industries.
But do talk a little bit more about it. Where do you think this is going to go? And maybe you could share some applications of what are you talking about, the biology on the chip that you built.
If you have a microscope, you can look at anything under the microscope, right? You can pretty much put any little molecule or cell or whatever and observe it. This is what we've done. We've created the creation or the innovation is a fundamental innovation. What that means is that it's profound in its sort of complexity and extent, that it's very broad in application. Now, just to explain it a little bit, how this works is that we took the fundamental sort of essence of biology, which is a molecule. We took that molecule and then we used it as part of a nanocircuit, right? So we take the molecule, we wire it into the circuit. The circuit itself is an amp meter, and then we apply a voltage across the circuit. And then what we do is that when the molecule is in place, think of it, it has a given resistance. Now, when the molecule interacts with its complement, so for example, if the molecule is an antibody and it interacts with its antigen, the overall resistance of the circuit changes. And because we are measuring voltage, current versus time, we can actually see these blips in real time when that interaction is happening. And so because we have then this circuit created, and it's an electronic circuit, it allows us to then deploy this on a microchip which is exactly what we've done. And then instead of having just one sensor and one molecule, suddenly you can jump to, thousands of sensors, to millions of sensors, eventually billions of sensors on these chips that are so low cost, but then the underlying technology is so much more powerful. That's where we are.
Yeah, so I would want you to do a little deep dive into it. You said, there's going to be broad applications, and I was on your website, and it said it could do from drug research and discovery to molecular diagnostics, DNA sequence, DNA
Yes.
digital storage, environmental surveillance tech. So maybe, if you could unpack a few of them to my audience, it would be like really great.
Biology consists of molecules interacting with molecules. Take one of the molecules, wire it on the circuit, I can detect the other molecule. That's the broadness of it. One area is that we were made to understand that in drug discovery, the ability to see, when you're developing a drug, you really need to see how this drug is interacting with its target to understand the probability of that drug being successful when you take it to clinical trials. And when we showed some of the drug developers the kind of signals we get, they were astounded that we can see one drug molecule interacting with its target at a single molecule level. This is a resolution that has never been attained. And so we're giving them the ability to see biology in ways they've never seen before, at the finest resolution. And what that is going to do for them is they are going to be able to look at these drug target interactions. They're going to be able to fine tune the drug to interact in ways they want, right? They may want it to have a given strength of interaction. In some cases, they want it a strong interaction. In some cases, it's a weaker interaction, and so on and so forth. And so this is going to be application number 0, where we're going to support that. At the same time, this is a general research tool, right? Anybody doing this sort of research of molecules interacting, again, looking at antibody antigen, looking at various proteins interacting with each other, looking at DNA probes interacting with each other. This is all in the context of research as well. If you take the same assay and put it on the diagnostic side, then it becomes a test, right? Suddenly, you can have a test that will do, say, COVID. But the power is going to be in that the ability to multiplex is tremendous, which means that instead of going to a doctor and saying, do you have COVID or not, the chip is going to look at all known viruses that infect humanity. I can have 100 viruses on one chip. And so, okay, the doctor is going to say, no, you're negative for COVID, but you have influenza, or you have RSV, or you have this other thing. And so you get a conclusive result. And so this is the future where I don't have to do one test at a time and the guesswork of, what's going on with me. And each test I take costs me so much more money and time. We need to have these chips where I can even run it at home. If it's the flu season, I run it weekly. I can run it daily. And I catch this information early and know that, oh my gosh, I'm sick. I don't even feel sick. But you catch it early, which means you're catching it when you're not showing symptoms, and you can intervene early. The disease is not going to ravage your body. The healthcare system is going to benefit from this because they are not admitting people. They catch disease early. The cost of care is much lower. The quality of life is much better. And we are going to stop pandemic spreading in our community because we catch them early. So, right, comprehensive testing, low cost testing, closer to the community testing is the disruption. Now, if I take the same sensor and I wire an enzyme in it, for example, the enzyme that makes a copy of DNA, which is the DNA polymerase, I can see what the enzyme is doing. This is the same sensor now being applied to sequencing. And this is the ability now to read bases, one base at a time. Because of the way this sensor works, it almost is observing biology in its natural state. So it allows the enzyme it allows us to read the sequence in real time, very fast. And because we have this ability to highly multiplex, we can see, we can have low cost, fast turnaround time on devices that are low cost. And this is what gets you to these sub $100 genomes. And then fast forward is all these sensors again. If you take them and put them in the environment, for example, I can monitor the environment for the protein on the surface of COVID, and suddenly you have an environmental sensor that can detect, these infectious agents in the environment, in the community. And then before you go to a location, you get an app that says, you get a notification on your phone and says, don't go to the supermarket because, we detected COVID in the environment. This is the future. And this is why I say the way we are testing and living today is so old school, and it's a limitation of the technologies. But now, finally, we're going to have these abilities and deploy these abilities in our communities.
You are saying that you are almost revolutionizing healthcare, right from your drug discovery to diagnostic to DNA sequencing. So far, I think most of our technology or tools were looking at band-aid solutions.
Yes.
But now we are completely re-looking at, redefining healthcare. The DNA, it's somewhat one of the most important thing. It makes us. It's got just four letters of the chemical ACTG, and you mentioned that it's almost just repeating letters, repeating itself. Deep down, if you're religious, if we believe that God created us, are we mechanical in nature somehow? Because when you're saying observing biology at its natural state, could you kind of unpack that a little bit from our audience, please?
The point I made about observing biology in its very essence is an important one, because a lot of the research we do today, right? You sort of want to freeze biology in a moment in time and then take that into the lab and study it, right? And a lot of what we're studying is in the context of we're trying to extrapolate the baseline state, and we're trying to extrapolate what is the meaning when I extracted this sample from that location. What does it mean? But what we're doing now is we are providing the tools to observe biology in real time as biology is happening. But we are observing it directly at a single molecule, one molecule at a time, instead of looking at a bulk reaction and then trying to understand what does this mean. This is the revolution. And what the impact of that is going to be, our knowledge and understanding is going to exponentially increase. And suddenly we are going to understand biology better. We're going to understand, and that understanding is going to result in us curing diseases like cancer, which has evaded us for all this time, because we can actually almost eavesdrop and watch biology and observe it in its very essence or natural state. And because of these tools that can record biology at this state, this is where the revolution is coming.
You mentioned about the application. Would you like to shine light a little bit on your Roswell DNA reader? Because you say that eventually, we will be able to store data into a DNA. Would you like to talk a little bit about that?
We did not even know this, but when we formed the company and started talking to other thought leaders, we were made aware that because of the way we store information today, each and every one of us, by virtue of having a cell phone, have actually created a data storage problem. And in the next 10 to 20 years, the data that is being stored will outpace the data storage capacity that can be produced. And so many of the companies that work in this space, the world is really looking for what is the next way or medium to store information. And like we always do, we go back to nature and we want to mimic nature. Nature stores information in DNA. That's how information about you is passed on from your grandparents to your parents to you, and we'll pass it to our kids and so on and so forth. We pass that information through DNA. And so the question is, can we store information in DNA? Today, we use binary letters, 0 and 1, to store digital information. DNA consists of 4 letters, like you said. Can we use the 4 letters or maybe just 2 letters to store that information? And so there's global efforts that are ongoing today to actually, one is write this DNA, which means if I have in digital information, say I have a book and I want to store that book in DNA, I need to write that and make a copy of that book as a copy of DNA, right? And there's projects where we've stored like music albums and books and that sort of thing, just as examples. Now, so you need to write the DNA. That becomes your little vial of information, and then when you need that information, you need to read it back, and that's the DNA reader that you need to read it. To make this commercially viable, obviously we can do both these things today. The question is, can you make it commercially viable to store digital information in DNA, which means you need the ability to write cost effectively, and you need the ability to read cost effectively, so that you can have these devices in your home. And this is where now a lot of the development and research is ongoing. Our reader, because of its ability to read in real time, very fast, right? We can read almost at the speed of the natural enzyme, and on devices that are low cost. We're on a path then to solving the read problem. And that's what we've been working on. And that's what you've been alluding to.
Right. Yes. So I'm going to go back to my earlier question, because I don't think I was able to articulate what I was trying to say. So there's this genetist, Andrew Hassel, who says that eventually we'll be able to code life. And I've been vested in virtual reality for a long time. And the far out dream of virtual reality is to create like a full dive virtual reality, and the intention there would be to kind of possibly copy the entire structural and functional data of the human brain, and possibly maybe like the DNA code of a human being. In these series like Westworld, do you think we would someday in the future will be able to do something like that, where we could possibly code life, create a simulacrum?
Well, that's a loaded question, and it's right. At the end of the day, I believe humans have souls, so I don't think it's just a physical copy of DNA that makes you who you are. And I think for many researchers and developers, usually there's a line you draw in the sand and say, how far can you go, right? Are you going to clone human beings, for example? Is that even ethical? And so I believe in these tools and technologies. There's things that are ethical, and I believe there's also things that border on being unethical as well. And my point is that we use these tools to improve life, to make us better, to fight disease, to figure out how we can increase our knowledge of biology and understanding of biology for the sole purpose of actually enhancing our lives, enhancing our well-being, right? Making us operate more optimally as human beings.
Fair enough. You have Mr. George Church on your scientific advisory board, and he's working on bringing the Wooly back to life. Would you have any views around that?
So I think and just to go back to the previous question, right? So sometimes we are creating these microorganisms where we are engineering them, but there's this point of you can use these processes for manufacturing, right? You can pull together different genes or different mechanisms in a cell, and then use that for manufacturing, right? So this is where manufacturing is going to move in the future, where you're using more biological mechanisms, right? So I believe that's something that's going to happen, and I believe it's going to be very helpful. It's going to be more environmental friendly, right? For example, there's biofuels that can be produced in some of these ways. The same mechanisms can also be used to actually, clean up pollution in our environment, potentially digest plastic, who knows? So there's other areas where we're moving to. And yeah, I believe some of the work that is ongoing, where they are starting with a copy of the woolly mammoth, and trying to, okay, can we clone that and bring back some of these extinct species. The difference is they are starting with a copy that exists, and I think it's a worthy project. And I think some of these animals failed or they got extinct because of the environment. Potentially things that we're doing and harming our environment are going to make many more species extinct. And we have a responsibility to protect our environment, to protect the species that live on this planet. And unfortunately, as humans, we've not done a good job of that. And we have a responsibility when we can to bring them back and create that environment that is more sustainable.
Lovely. Thank you, Paul. Really appreciate you taking time and being part of the podcast. Would you like to share what applications you are most excited? What do you think are going to come out of Roswell ME Chip?
I am really excited. I like to live at the interface of the development and its application in the community. I look forward to the day where we'll have, testing being informed by knowledge, where you get a text on your phone and says, hey, there's something going on in your neighborhood, be on the lookout for this, run this chip every week, and that sort of thing. So sensors in the environment, sensors testing in the home, a knowledge base that drives that. And I believe bringing this closer into the community, and also bringing it in a way that is going to drive accessibility so that everybody can have access to it, not just a few who are rich and wealthy, but everybody can have access to it. Everybody can have access to their own genome. Everyone can have access to tailored therapeutics and precision medicine. And this is the future that makes me exciting and gets me out of bed to drive this innovation into the market.
Thank you. Really, really appreciate you taking the time, and I wish your dreams come true. Listen to my listeners. If you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you. Really
Absolutely, Eddie. It's a pleasure, and keep doing the good work you're doing. And I look forward to continuing these conversations.
Thanks a lot. I really appreciate this.